Hi, welcome to Pathology Riddles. Today we are going to deal with this topic called as mechanisms of cell injury. Last week we spoke about what are the causes of cell injury and how a cell responds whenever it is exposed to an injurious stimuli or stress. The first mechanism is hypoxia and ischemia. So when does hypoxia and ischemia happens? The most common cause is when the blood vessel is narrowed or blocked by a clot, especially by this disease called as atherosclerosis. So when the blood supply reduces to that particular organ, what happens? The cells of the organ will not have enough supply of oxygen to conduct this oxidative phosphorylation. So the oxidative phosphorylation is going to decrease. And if the oxidative phosphorylation decreases, the amount of ATP generated also decreases. What all happens when there is decreased ATP? First of all, the sodium-potassium pump and the calcium pump get affected. They require ATP for their smooth functioning. So what is the function of the sodium-potassium pump? It usually takes sodium out of the cell and potassium inside the cell. But here what happens is the sodium starts accumulating inside the cell because of the pump not functioning properly. So as a result, when sodium is in excess, then the water will also increase inside the cell. And the potassium which is required to come inside keeps getting out. So because there is increased sodium and water, there will be swelling of the cell. There will also be swelling of the endoplasmic reticulum and the microvilli which maintains the structure of the cell will also disintegrate. And because of the excess fluid accumulation, the cells will form projections and they are called as mbbs. On the other hand, even calcium pump will not work properly. So more and more calcium will get deposited inside, and increased calcium deposition in the mitochondria can lead to damage to the cell. Secondly, because the oxidative phosphorylation is not happening properly, that can increase reactive oxygen species, which are another cause of cell injury, which we will deal in the next part. So the third thing that happens is when there is not enough oxygen, then the cell will start doing anaerobic glycolysis, that is breakdown of glucose in the absence of oxygen. As a byproduct, there will be more and more of lactic acid. The glycogen will be used up in this anaerobic glycolysis, so that will reduce. More and more of lactic acid will form, and that will decrease the pH, which will inhibit or affect the enzyme function. And the next thing that happens when our decreased ATP occurs is ribosomes get detached. So because of these four main mechanisms, that is increased reactive oxygen species, then cellular swelling, decreased pH affecting enzyme function, and ribosome detachment, all these are going to finally result in membrane damage, and finally the cell will undergo cell death by necrosis. So that is about the first mechanism of cell injury, that is hypoxia and ischemia. Now we go to the second mechanism of cell injury, that is oxidative stress. So what do you mean by oxidative stress? They are cellular abnormalities that are induced by reactive oxygen species. So what are reactive oxygen species? They are molecules that belong to this group called as free radicals. They have a single electron in the outermost orbit, so they are very unstable. So the reactive oxygen species belong to free radicals, which are very unstable molecules, and the cellular abnormalities that are caused by this ROS or reactive oxygen species is called as oxidative stress. So what causes this production of reactive oxygen species? They could be caused by chemicals. They could be caused by radiation. Even hypoxia or decreased oxygen can cause the production of reactive oxygen species. Even ischemia reperfusion injury, that is when the blood supply is less and then we try to reperfuse that area, that is increase the blood supply or increase the oxygen, that time there can be more production of reactive oxygen species. Even cellular aging can increase the reactive oxygen species production. And finally, inflammatory cell-mediated injury. As we all know, inflammation is actually protective. It is a vascular response to cell injury, but sometimes inflammation can also result in injury. So that is by the production of reactive oxygen species. I hope you understood what is oxidative stress and what are the causes of the production of this reactive oxygen species. And this is a very important question asked usually in almost all the exams. So let's know more about these reactive oxygen species. So the first reactive oxygen species is superoxide. So how is superoxide produced? It is depicted as O2 minus. Mechanism of production of superoxide is they are produced during mitochondrial oxidative phosphorylation because of the incomplete reduction of oxygen. They are also produced by the WBCs and they have this enzyme called as phagocyte oxidase which produces this superoxide. And how is it removed by our body? By conversion to water and oxygen by this enzyme called as superoxide dismutase, which is already present in our cytosol. The next reactive oxygen species is hydrogen peroxide. And how is this produced? It is produced from the previous free radical, that is superoxide, by the action of the enzyme called as superoxide dismutase. And how is it removed from the body? How is hydrogen peroxide removed from the body? By conversion to water and oxygen by two enzymes. One is catalase and other one is glutathione peroxidase. The third reactive oxygen species is hydroxyl radical. This is also produced from water, hydrogen peroxide and superoxide. And it is removed from the body by conversion to water by glutathione peroxidase. And lastly, we have peroxynitrite. The mechanism of production of peroxynitrite is by interaction of superoxide and nitric oxide by nitric oxide synthase enzyme. And peroxynitrite is removed by conversion to nitrite by enzymes in mitochondria and cytosol. So all these things that are removing these particular reactive oxygen species are called as antioxidants. Along with these enzymes, there are also these vitamin A, vitamin C, vitamin E antioxidants which try to remove all these free radicals continuously. And we all keep talking about stress most of the time. It is caused by this. And in times of stress, these free radicals are increased. And this is called as oxidative stress. When free radicals increase, it is called as oxidative stress. So how can these reactive oxygen species of free radicals cause damage to our body? They can cause lipid peroxidation. The lipid which are present in the membrane can undergo peroxidation and that can damage the membrane. They can cause modifications in the proteins which can break down the proteins or cause them to misfold. And then finally they can also damage the DNA leading to mutations in the DNA. So the free radicals cause all these changes in our body and that's how they cause oxidative stress. So that completes oxidative stress. Next we have this mechanism called as ischemia reperfusion injury. This mechanism is actually supposed to be protective. That is, whenever there is decreased blood supply, if the blood supply is restored, it's supposed to protect the cell from damage. But sometimes in cardiac damage or in the cerebral ischemia, there can be ischemia reperfusion injury. When we are restoring the blood supply, there can be more harm done than good. Now, what are the mechanisms that cause damage when there is reperfusion after ischemia? One is production of reactive oxygen species. So what are the mechanisms of production of reactive oxygen species? As we know that some kind of damage has happened. So in that damage, the mitochondria of some cells are damaged. So there is incomplete oxidation, and that results in reactive oxygen species. Second, because of the damage, the antioxidant, that is the enzymes that remove the reactive oxygen species, are compromised or they are reduced. So as a result, there is more of increased reactive oxygen species. And third, whenever there is damage, the body responds by inflammation. So the inflammatory cells will reach that area and they will also produce reactive oxygen species in the process of removal of damaged tissue. So all these are going to increase the reactive oxygen species, which can result in damage to that tissue which has already undergone ischemia. Second is activated complement system and inflammation products. These two will also get activated whenever there is ischemia reperfusion injury. So complement system, finally the end product, can cause damage to the cell. And then the inflammation products which are formed, they can also cause damage to the cell. So these are the ways by which ischemia reperfusion injury occurs. The next mechanism of cell injury is by toxins. Toxins can cause cell injury by two mechanisms. First one is directly acting toxins. So an example of directly acting toxins is mercury chloride poisoning. How do people get mercury chloride poisoning? By ingestion of contaminated seafood. So what does this mercury chloride do? It binds the sulfhydryl groups of various membrane proteins. And once it binds, what it does is inhibits ATP dependent transport and also increases the membrane permeability. Both can result in cell death. The second type of toxins is latent toxins. They themselves will not cause harm, but they will be converted to a toxic radical which will cause harm. Like for example, carbon tetrachloride is converted to toxic free radical in the liver in the smooth endoplasmic reticulum by P450 enzyme. And this toxic free radical will cause cell injury by membrane peroxidation, and we know it can damage the proteins as well as it can also cause damage to the DNA. So this mechanism is how toxins cause cell injury. The next mechanisms of cell injury is endoplasmic reticulum stress. Normally, a step in protein synthesis is protein folding. Sometimes the protein can undergo misfolding. As soon as misfolding happens in the endoplasmic reticulum, and when it is trying to release it into the cytoplasm, the endoplasmic reticulum membrane will have some sensors which will identify these misfolded proteins. And when they sense this, they are going to signal the chaperones. The chaperones are the checkpoints which will correct this protein response. And how they are going to do it? They will increase the production of chaperones. They will decrease the protein synthesis so that more and more misfolded proteins don't occur. And whichever are produced, increased protein degradation occurs. So that's how we reduce the load of misfolded protein so that they cannot cause any injury to the cell. This is how normally we function, that is the adaptive unfolded protein response occurs. Whenever there is misfolded proteins, the chaperones will increase, there is increased protein degradation and there is decreased protein synthesis. But what happens if the misfolded proteins are produced in very large quantity, then there will be activation of this pro-apoptotic proteins, which are BH3, and there will be activation of caspases. So BH3 are sensors which will activate the pro-apoptotic proteins, and then finally the pathways of apoptosis will activate the caspases, and finally there will be terminal unfolded protein response, also called as apoptosis. So how does endoplasmic reticulum stress cause cell injury? The protein misfolding that happens within cells, it can cause disease by creating a deficiency of an essential protein. Because they are misfolded, so final protein is not produced. And then by inducing apoptosis, as we saw the unfolded protein response, whenever there is excess of misfolded proteins, they undergo apoptosis. Just like in the proteins, we have got chaperones, which is the guardian of the misfolded proteins. In DNA damage, whenever there is any damage, we have something called as p53 proteins. So whenever there is any damage during the mitotic cycle, it is sensed by this p53. It will arrest this mitotic cycle at G1 phase and then try to repair the DNA. Suppose the repair is not possible. If the DNA damage is irreparable, the p53 will signal apoptosis by activating the pro-apoptotic proteins. And lastly, the final mechanism of cell injury is inflammation. As we already saw in ischemic reperfusion injury, the inflammatory cells can secrete products to destroy the microbes. But these products can also damage host tissue. So the mechanisms of cell injury that we have studied today are hypoxia and ischemia. So the second one was oxidative stress. The third one was due to toxins. It could be latent toxins or directly acting toxins. The fourth one was endoplasmic reticulum stress or misfolded protein response. And then we had DNA damage, which are checked by the p53, and damage caused by inflammation. I hope you have understood all these mechanisms. In case you have any doubts, please write in the comments so that we can answer them. This is Dr. Susan signing out till we meet in the next video.